Let's continue the special senses by looking at hearing and balance. Now remember, the ear has two special senses associated with it, not just hearing but balance also. So the ear itself has got three different regions. First is the external ear. That goes from the fleshy outside, which is actually called the auricle, which is what you think of your ear. Then you got this external auditory meatus, which is this passageway leading back to the tympanic membrane, which is your eardrum. Deeper to the inside is the middle ear region, filled with three little bones. These tiny little auditory ossicles are all about sound amplification. Then you got these structures deeper within the inner ear, all about hearing and balance. And there's many things there we'll take a look at. But looking back at that external ear, again, the outer fleshy part is called the auricle or pinna. That's cartilage there primarily. Of course, you got skin and epithelial tissue over the surface. Deeper, there's a passageway called the external auditory meatus. It's what a meatus will always be a passageway of some kind. And there'll be hairs and ceruminous glands to help protect that passageway, hopefully keeping structures out of it, maybe tiny bugs or something like that. But again, it terminates back at your eardrum, which is the tympanic membrane, a very thin, sensitive membrane that'll move when sound waves hit it. But deeper to the inside, there's the middle ear region. Now, it's separated from the inner region by oval and round windows we'll look at further along. There's also passageways for air leading into the middle ear region. That way you can balance air pressure on your eardrum. If you ever get pressure on that eardrum one way or the other, positive or negative, it'll be pushing on it and it won't vibrate easily and you won't be able to hear the way you should. But if you look at our throat, which is our pharynx, there are these auditory or eustachian tubes that lead back into this middle ear region. Whenever you yawn or swallow, skeletal muscles will open them, and that lets air move back towards that middle ear region to balance the pressure, whichever way it needs to move. You also have from the mastoid process, which remember is that big bump below your ear, you probably saw that back with the bones, a passageway to these mastoid air cells. But these three bones in your middle ear are called the malleus, incus, and stapes. And again, they will move and amplify the sounds that you hear. And that gives a large sound amplification. Looking deeper to the inner ear, there's structures like the semicircular canals, vestibule, and cochlea we'll look at a little further. But there's also some labyrinths or chambers, and you have these bony chambers in the temporal bone, and then you have these membranous chambers back further inside the bony labyrinth. But they're also filled with lymphs, fluids. Whenever you hear about lymphs, that's what they will be. Endolymph and perilymph are the two you'll find here. The endolymph is found inside the membranous labyrinth, and the perilymph is a space between the membranous labyrinth and the periosteum of the bony labyrinth. Looking at some other structures like the oval window we mentioned before. That's a membrane leading from the middle ear to the inner ear. It communicates with the vestibule, which is a little chamber over here shown in the picture, which communicates with the scala vestibuli of the cochlea. We'll look at that a little bit further along. Cochlea here is what looks like a little snail shell. That scala vestibuli extends from that oval window to the helicotrema, which is the apex or the end of that cochlea. That second cochlear chamber, the scala tympani, leads from the helicotrema to the round window, another one of those structures seen in this region. And these areas are filled with the perilymph. Looking further at that inner ear, looking at the wall of that scala vestibula, which is in the cochlea, there's a vestibular membrane. And that membrane inside that cochlea separates the cochlear duct from the vestibular duct, and again it's filled with endolymph. The wall of that scala tympani, located in the helicotrema, is the basilar membrane. That's what the wall of it will be. And that separates that scala media from the scala tympani. But looking back at that cochlear duct, this is a space between the vestibular and basilar membranes seen above, filled with endolymph. Now the width of that basilar membrane is very important. You can see that it has a varying width right here. And down here at the bottom you can see it increases from 0.04 millimeters near the oval window to 0.5 millimeters near the helicotrema. Now back near that oval window, near that basilar membrane, this is the region that responds to high frequency sounds. Near the helicotrema at the opposite end is where you hear low frequency sounds. Looking further at the inner ear, we have this spiral organ. Here you have cells in the cochlear duct. These little epithelial cells along this spiral organ allow for the creation of action potentials, electric signals that we'll interpret at the brain as what we hear. It contains hair cells with hair-like projections at the apical ends, and even though these are microvilli, they're actually called stereocilia. The basilar region, which separates the scala media and the scala tympani of those hair cells, is covered by synaptic terminals of those sensory neurons. And the cell bodies of the afferent neurons, remember afferent and efferent's been discussed before, are grouped into cochlear ganglion. And remember, a ganglion is a collection of neuron cell bodies outside that central nervous system. And all those afferent fibers are going to form that cochlear nerve. It's going to be all the axons coming off those neurons there. Back up here at the top, we see those hair cells, which detect movement. Now, there's inner hair cells, which are responsible for hearing, and there's outer hair cells, which regulate tension on that basilar membrane we mentioned before. There's a hair bundle where we have stereocilia of just one of these inner hair cells, and attached to them are tip links. They call these gated springs. These little proteins look like a coiled spring, and on the other end of it, it's actually attached to the gate of potassium channels. So movement will actually open those gates, allowing for the movement of potassium in the creation of action potentials. Not often you see mechanically operated gates, but here in the ear we do have them. So what has to happen for us to hear something? First of all, the air around you has to be moved. That's what we think of as a sound wave. It's going to strike that tympanic membrane, causing it to vibrate. That vibration of your eardrum causes those three bones in the middle ear to move themselves. That amplifies the sound. The foot plate, which is at the very end of that last bone, the stapes, will move the oval window. The vibration of that foot plate will cause the perilymph on the other side of that oval window to move in that scala vestibuli. As the stereocilia in that region are bent, the potassium channels are opened, and action potentials are generated and sent back to the brain. And somehow the brain interprets that as all the different things that we hear. There's also some muscles in that middle ear region too, like this tensor tympani, which attaches to the malleus, and the stapedius, which attaches to the stapes. And what these muscles do when there's very loud noises, they pull on those bones, and it keeps them from moving, which keeps them from amplifying sound. And that'll keep very loud noises from damaging the very delicate inner ear structures. And of course, those are operated automatically, autonomically, and that's what's called the attenuation reflex. But looking back again at that basilar membrane, remember the very high-pitched sounds are perceived close to that oval window. That's back at the beginning of the cochlea. The low-pitched sounds are perceived further away near the opposite end of it. But looking also in this inner ear region, remember, we also have little sensoreceptors that are not only there for hearing, but also for detecting movement. And that's where we get the static labyrinth and the kinetic labyrinth. Now, looking at the static labyrinth, that tells us head position. That way we always know which way is up. There's this region called the macula inside the vestibule with the little specialized epithelial cells, which are actually columnar cells, and hair cells with numerous stereocilia. Now, on them are one cilium. They're embedded in a gelatin-like material, so it's not liquid, not fluid, but something thicker. And inside of it are tiny little bones called otoliths, and those literally mean ear bones. Whenever that gelatinous mass moves as you tilt your head one way or another, gravity pulls on that material. That will move those hair cells, and the movement of those hair cells will create action potentials, which are interpreted by the brain as which way is up. But looking at that kinetic labyrinth, this detects movement, right? Which way is up is one thing, but movement's another. This happens inside those three semicircular canals filled with endolymph. And if you look at those canals, you'll notice that there are three of them running in three different directions. So that's going to give us three different planes, a transverse, coronal, and sagittal. All that's been covered as far back as chapter 1 in your book. The base of each is expanded into an ampulla, with little sensory epithelia called crista ampullaris. At those, you'll find little cupula, which actually look like tiny little upside-down inverted cups suspended over these little hair cells, which notice act as a float. And whenever they're moved, in other words, displaced within these semicircular canals, electric signals, action potentials will be generated, and this system right here detects changes in the rate of movement rather than just movement alone. And talking about the effects of aging, when it comes to odors, there'll be a slight loss at first, then a little bit more as the years go on. You'll lose a little bit of taste, and remember, smell and taste actually go together, working together. You'll lose the flexibility in the lens. That's usually when you'll need reading glasses because you'll have trouble focusing on objects closer than 20 feet. You may get the development of cataracts. Cataracts is a buildup of the proteins, causing a clouding of the eye that scatters the light rays and you can't see as well. Macular degeneration just refers to all the natural effects of aging on the eye, so that refers to many things. Might get glaucoma, a buildup of pressure in the eye because those little canals of schlemm have gotten blocked with proteins. The fluid can't get out, the pressure builds and it distorts your vision. And with diabetes decreasing blood flow to things around the body, the retina's one. You could lose enough blood flow to your retina, you could go blind, and it happens quite often. And of course, you'll lose your sharpness and vision, color perception, and everything right along with it.